Hello, bachcho. Let's see the question. So the question says the stickiness of DNA ends facilitate the action of which enzyme? So इन चारों ऑप्शन में से हमें आइडेंटिफाई करना है कि स्टिकी एंड्स कौन से एंजाइम को हेल्प करता है. So the key concept in this question is to first understand what are the sticky ends in DNA. So the sticky ends in DNA are formed due to the cleavage of DNA by restriction enzymes. So when the restriction enzymes cut the DNA into two fragments by the help of staggered cuts, then that results in the formation of sticky ends. So for example, let's consider the recognition sequence for the enzyme echo R1, which is also a restriction endonuclease. So if this is the recognition site for echo R1, then the cleavage of the DNA occurs at this position and at this position. So therefore, we will get two fragments of DNA as follows. So this will be the first fragment of DNA and this will be the second fragment of DNA. So now we can see some overhanging nucleotides present. So this is the flanked region or the overhanging nucleotides, and this is also the flanked region or the overhanging nucleotides present at the end of the both fragments of the DNA sequence, which is cleaved by the enzyme echo R1. So therefore, these flanked overhangs are known as the sticky ends. So these ends which are present in the DNA fragments due to the cleavage by restriction endonucleases are known as the sticky ends. So if we see now one of the fragments, then this is the one fragment. So now this is the sticky end or the overhanging flank present in this DNA fragment. Now this overhanging DNA flank can help in joining this DNA fragment with another DNA fragment which has a complementary sequence to this sticky end. So therefore the DNA fragment having the overhanging flank with the sequence as follows can bind with this sequence. So since this sequence is complementary to this sequence, therefore these two ends can join together to form a single sequence of DNA. So now the joining of these two fragments is done by the enzyme DNA ligase. So we can say that the sticky ends of the DNA fragments facilitates the enzyme DNA ligase. So the stickiness of the DNA facilitates the function of DNA ligase for joining the two fragments of the DNA. So therefore, if we see the enzymes given over here, then the first enzyme, that is the first option given, is DNA polymerase. So DNA polymerase is the enzyme which is responsible for the synthesis of new strand of DNA, and it is not responsible for joining the sticky ends of the DNA. So therefore, this won't be the answer. The second option says DNA ligase. So this will be the answer because DNA ligase is the enzyme which is responsible for joining of the two DNA fragments, and the sticky ends help the DNA ligase to join the complementary strands together. Now the third option given is restriction endonuclease. Again, this won't be the answer because restriction endonuclease cleaves and forms the sticky ends and not facilitates. So the sticky ends does not facilitate the action of restriction endonuclease. Now the fourth option says alkaline phosphatase. Again, this is incorrect because phosphatase enzyme is responsible for addition of phosphate group and is not in any way responsible for joining of the two DNA fragments. So therefore, the final answer for this given question is option number 2, that is DNA ligase, as the enzyme which is facilitated, whose function is facilitated by the stickiness of the DNA. So this is our final answer. Hope you understood this well. Best of luck. Thank you.